And here we are back with the last topic, that is chapter number 7. So starting with the subtopic 7.1, DNA and RNA structure and function. So we'll talk about these. First of all, you should know that DNA stands for what? So DNA stands for deoxyribonucleic acid, while the RNA stands for ribonucleic acid. So this is how we do it. KPI number 1, recognize that DNA and in some cases RNA is the primary source of the heritable information. It means that the DNA can transfer from one generation to the next generation, and it carries all the information, all the genes on it. Which row in the table below provides the correct sequence or source of heritable information for each listed organism? So we need to see like what happened. So again, apply the same rule that target anyone and then you can go with it. For example, I want to target virus, okay, and I know that in the viruses, as per my knowledge, DNA or RNA can be the genetic material, and not RNA only, not RNA only, not DNA only. So it means that the 2 is the correct answer, that is B over here. So this is the only correct answer we have. It's so simple, so easy. This is called as elimination rule that you have to follow. A scientist studying previously undiscovered virus determined that its genetic material had the following composition of adenosine 16%, guanine 37%, thymine 0%, and cytosine 24%, and uracil 23%. Which of the following correctly describes the viral genome? So basically, when you have uracil, it means that the genome is RNA based. Since thymine is 0, thymine is present in the DNA. This is one of the difference between them. So we can easily say that the viral genome is single-stranded DNA. No. So it's either RNA single-stranded or double-stranded, and we know that the RNA is always single-stranded. Describe using visual the double helix structure of the DNA. This confirms us that RNA is single-stranded while DNA is double-stranded. The 2 strands of the DNA, they look like this. They are twisted around each other. They run anti-parallel, and the basic unit of both DNA and RNA is nucleotide. It's nucleotide, and each nucleotide has 3 components. So if we draw the nucleotide structure, it looks like this. So this is a ribose sugar, and on one side of the ribose sugar you have adenine, guanine, cytosine, or thymine, and that's why we say that this side is nitrogenous base, while on the other side we have phosphate group. So we have ribose sugar, we have the 3 components including nitrogenous base and the phosphate group. Use the diagram. One difference between the DNA and RNA in this structure is this 2 group. If you have OH over here, this is RNA nucleotide, and if you have only H over here, this is deoxyribonucleic acid, because we remove the oxygen, so it is DNA nucleotide. Use the diagram below which shows a part of the DNA molecule to answer question 74-76. Identify the structure labeled with letter X. So first of all, this is X. So in X, what do you have? You have a nitrogenous base, you have the sugar, and you have the phosphate, all 3 together. So it is a nucleotide. And list is 3 components we have done already over here: phosphate group, nitrogenous base, and ribose sugar. Identify. So here number 1, phosphate group. Number 2, ribose sugar. Not ribose sugar, you should say deoxyribose sugar, because it is DNA. Deoxyribose sugar. And number 3 is nitrogenous base. Okay, nitrogenous base. Moving to next, identify the label that represents purine. Now over here we have nitrogenous bases are of two types. So if we go to nitrogenous bases, so you should know that they are either purines, which are A and G, or they are pyrimidines which are C, T in the DNA, or they are U in RNA. Okay, so make sure that you know about this one. And purines are double ring nitrogenous bases. So you have like this, so one ring and then you have another ring which goes something like this, while the pyrimidines are single ring structures like this. Okay, identify the label that represents purine. As we said, purines are double ring, so double ring, you need to see the labels. So it's either this C or this A, so of course double ring is A, so it's a purine. Identify the chemical bond labeled Y, so we need to see the bond. This is the dotted bond over here which is connecting the purine and pyrimidine, so this is a hydrogen bond. And how this is important to the DNA structure, because it connects or it joins the nitrogenous bases together. Use the diagram to answer 77 till 79. Identify what letters D, H, X and Y in the diagram represent. So I will just label it on the top. Y is a phosphate group. Y is phosphate. D is deoxyribose. Then H is a bond. It's phosphate. No, it's a how you will say it actually. It's the bond that is connecting the ribose with the phosphate, so it will be covalent bond. It will not be a phosphodiester bond because we are not talking about the whole chain, so we are just talking about one bond, so it's a covalent bond. And X is hydrogen bond, so hydrogen bond. Okay, name the nitrogenous base that are represented by N1, N2, N3, N4. I will also label it on the top. You should know that A comes with T, T comes with A, N3, G comes with C, and C comes with G. So this is how you will answer them. Explain the double helix, which is being used to describe the DNA structure. We discussed in the previously, it means that DNA is made up of the two strands which are complementary to each other, and they run anti-parallel and they are twisted around each other. That's why we call it as a helix. Which of the following correctly describes the structure of the DNA? DNA is single-stranded? No. DNA is made up of the two identical strands that do not interact with each other? No. DNA double helix is formed by a covalent bond between? No. DNA consists of the two complementary strands twisted around each other, and that is the answer for here. You can write the same up to get the full marks. Describe the role and number of hydrogen bonds. We discussed already the role. It connects the bases together. And what is the number? How you calculate the number? So A always joins with T with 2 bonds, while G always joins with C with 3 bonds. So it's so easy. Just count the nucleotides and you can find the number. Which of the following is correct about the two segments of the DNA shown in the diagram below? Segment 1 belongs to prokaryotes. How it can belong to prokaryotes? It's like a DNA, so it's wrong. Segment 1 cannot code for mRNA. It's wrong. Segment 1 contains deoxyribose. Will segment 2 contain ribose? No, since both the segments are of the DNA, so no. Segment 1 has less hydrogen bonds while 2 has more. That's possibly correct, and we are only left with this one. So what we can do, but we can count the number of the bonds. So 2, 2, 2, 2, 3, 2, 3, 2, 2, 3, 2. So you can add them. 2 plus 2, 4, 6, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. So here we have 25. Okay, here 3, 3, 3, 3. So 12. Then again 3, 3, 3, 3. So again 12. So 12 plus 12, 24, 25, 26, 27, 28, 29, 30. So here we have 30. So what it says, segment 1 has less hydrogen bonds, 25, 2 has more hydrogen bonds, 30. Yes, that's correct. Segment 2 requires high temperature to be denatured. Yes, if you have more hydrogen bonds, you need more temperature to break them. And how you see more hydrogen bonds? If C to G are more, it means more hydrogen bonds, it means more temperature is needed. So contains deoxyribose, 1 has more purine bases, 2 has more hydrogen bonds, 2 has more pyrimidine. So this is correct. How many hydrogen bonds does the DNA segment has? You can easily calculate 2, 2, 2, 2, and 2. So 1, 2, 3, 4, 5, 10. And I will do the 3 up. 3, 3, 3, 3, 3, 3, 3. So 1, 2, 3, 4, 5, 6, 7, 21. So the total 21 plus 10 is 31. So 31 is the answer. Okay. Moving to next. Which of the following correctly describes the typical flow of the genetic information in the cells according to central dogma of life? This is the central dogma. Okay. This is called central dogma of life. Central dogma of life. And what does it say? It says that DNA can make DNA. So this is how it goes. DNA can make DNA, and this is called as replication. While when DNA converts into mRNA, this is called as transcription. And when RNA converts into protein, this is called as translation. Now, the RNA can go back to DNA, so it is reverse transcription. Now, since we are over here, we can quickly discuss about the role of the enzymes on the top of the same diagram. Replication, you mainly need DNA polymerase. The most important enzyme is DNA polymerase. Of course, you need DNA helicase and also DNA ligase over here, and DNA topoisomerase also, which will control the over twisting. Okay, so topoisomerase. But when you do the transcription, the main enzyme you need is RNA polymerase. Okay, and when you do the reverse transcription, you need reverse transcriptase enzyme. Okay, and translation takes place on the ribosomes. You need tRNA and you need rRNA for this process, and tRNA is the one that carries the amino acids, and anticodon. Okay, so this is how it works. Protein is made up of amino acids. And how it is made up of amino acids? Because these amino acids join together, by peptide bond. So this is how you say that they join together by a peptide bond. We will now go back to this question. Which of the following correctly describes a typical flow? We know the typical flow, that it's proteins store genetic information and direct the synthesis of DNA and RNA. RNA is directly replicated. No. DNA is proteins encode RNA, which is then used to synthesize DNA. No. DNA is replicated, then RNA, then transcribed to RNA. Yes. Then translated to protein. Yes. So this is the correct answer. Identify the processes. We did already. So identify the process 1, process 2, process 3. You can see on the diagram, I have already done it. Recognize. Oh, we are done with first topic. Let's move to the second topic. That's replication. Replication means making copies. Okay, so copying the DNA. And which model is used by copying the DNA? It is semi-synthetic conservative conservative replication. So the method that is used over here, it is semi-conservative replication. Which of the following is the correct direction of the DNA? So please remember the direction is always 5 prime to 3 prime, 5 prime to 3 prime direction. RNA is also made in 5 prime to 3 prime. Not this one, not this one, not this one. Okay. Differentiate between DNA replication in the prokaryotes. So this is the one in the prokaryotes. It's a loop shaped DNA, and the new DNA is made from the original DNA. Replication fork is made, which is only one, and the same enzyme DNA polymerase is used over here. But if you look into the process B, which is in the eukaryotes, so when we talk about the eukaryotes, you see many replication forks are made. So replication bubble, replication forks are made many, or you can say as multiple. It's not only one. New DNA is made from the original DNA. Identify the process A. So process A is replication in prokaryotes, while process B is replication in, so this is replication in eukaryotes, while this one is replication in prokaryotes. Okay, moving to next. Identify one difference. We already said that major difference is that it shows the origin of replication, which is only one, and it has many or multiple origin of replications. Complete the table below. Compare and contrast replication. So number of origins, 1. Okay, and DNA replication in eukaryotes, it is multiple or many. Okay, location is in the cytoplasm, while this one, it is in the nucleus. Number of the DNA molecules, so of course it's 1, and over here we have many. And the shape of the DNA is, we saw up, it's circular, and here it is linear. Next topic, 7.3, transcription and then RNA processing. We already know that transcription, DNA to pre-mRNA. If you find the pre-mRNA in between and then you go to final mRNA, this process is called the RNA processing. Okay, and this is called as transcription when DNA converts into RNA. Describe using the details of the process of the transcription. Include the role of the RNA polymerase. We know that RNA polymerase will join the new RNA nucleotides and to the developing strand. Template and non-template coding DNA strands. Non-template is called as the coding DNA strand, and the role of the promoters, which are the places where RNA polymerase bind. So at promoter, this is the place where RNA polymerase join on the DNA, and terminators are the one which will give the signal to terminate the process. Which of the following best describes the function of the RNA polymerase in the replication? So it unwinds the DNA, reads the template strand, and synthesizes the new RNA strand. Yes. It binds to the ribosomes. No. It removes the introns. No. It copies the entire DNA. No. It only copies the non-template or the coding strand. So it's the answer. During transcription, how do the template and non-template, which is the coding strand, function? Okay, the non-template strand is read by the RNA polymerase, while the template strand is used as a reference. No. Until here it is correct. Okay, RNA polymerase reads the template strand. Oh no, upper one is even wrong from here. Okay, RNA polymerase reads the template strand to synthesize RNA, while the non-template strand has the same sequence of the resulting RNA. Okay, that makes sense. Both DNA strands serve as a template. No, the non-template strand is transcribed into RNA, while the template strand remains inactive. So this is also wrong. So B is the only correct. What is the primary function of a promoter? Promoter, as we discussed up, it is a place where RNA polymerase binds. So let us see how it happens. It signals the RNA polymerase to stop. No, that's the terminator. It codes the first. No, that's the translation. It splices the newly synthesized before translation. No, that is role of spliceosome. DNA region where RNA polymerase bind to initiate transcription. Yes. Okay. From here, initiate means the step one. Okay, step one, initiation. Step two is elongation. Step 2 is elongation, when RNA polymerase will add up more nucleotides. And the last one is termination, when the terminators will give the signal to end transcription. How do terminator sequence contribute to transcription? This is how I told you. They will cause the RNA polymerase to separate from the DNA and result in the end of transcription. They recruit ribosomes? No. They guide RNA polymerase to the promoter? No. They cause the RNA polymerase to detach from the DNA, signaling end. Yes, this is one. They convert the pre-mRNA to mature mRNA? No. So this is what we said. This is the role of the spliceosomes. A transcribed region of the DNA has the following sequence: 5' GC 3'. Which of the following? You know that it will be made in 5' to 3', right? So we need to see over here 5', then G, then which of the following represents RNA sequence? So we need RNA sequence. So A is T, there is no T, so it is U, then C, then G, then C, then 3'. Okay, look for 3' to 5' like this one. So 3' starting with C, A, no, G, no. So C, G, C, U, G. So last one. Okay, so it matches with that. The template strand of a gene contains the sequence 3', this one. Write the non-template strand of the sequence and the mRNA sequence. You need to specify the 5' to 3'. Okay, so let us write the non-template strand for this one. So first of all 5' and then A, so it is T. If it is T, then it is A, G, C, A. Moving further, then it is T, C, G, A, T, and 3'. Now we will do RNA. So 5', it's T, so there will be no T, it will be U, A, G. So it will be C, A, U, C, okay, C, G, A, U, 3'. Okay, so this is how it works. Describe the main steps of the transcription. So we discussed already. Initiation is a process in which RNA polymerase bind to the promoter region. Okay, it causes the unwinding and then finds the promoter region to bind over there to start the transcription. Elongation is when RNA polymerase elongates by adding the new nucleotides, and termination when the terminators cause the detachment of the RNA polymerase to end transcription. Explain using visuals how RNA is processed after transcription. So they say visual, it means you might have diagram or you might not have diagram. Okay, include the addition of the poly A tail, number 1, addition of the GTP cap, number 2, RNA splicing, number 3, and alternative splicing, number 4. But in this RNA splicing, basically what happens in this RNA splicing? Introns removed, okay, and exons join back. Okay, so remember this one. So this is actually, that is 3 and 4. Okay, so 3 and 4. So this is 3, this is 4. So these are the 4 things that happen in the RNA processing. The white gene of the fruit fly, it contains 5868 base pairs long. However, the final mRNA is 2352. Which of the following best describes why an mRNA transcript can be shorter than the gene which was transcribed? You know that there will be post changes, and also the gene doesn't contain all the template part. So the gene contains segments which are like limited to be transcribed. RNA transcripts are single-stranded, therefore smaller. No. The non-template strand of the DNA contains additional sequence. No. The gene contains segments of the sequence that have been removed. Who is removed? Introns, right? Yes. The RNA nucleotides are smaller than the DNA nucleotides. No, it's not about the nucleotides. A transcript unit that is 8,000 nucleotides may use 8,000 then 1,200 nucleotides to make a protein consisting of 400 amino acids. The best explained by the fact that, okay, many non-coding stretches of the nucleotides are present in the transcribed RNA. Yes, that's possible. There is redundancy and ambiguity. We didn't study the properties of the genetic code, so these are the properties. Many nucleotides are needed. No. Nucleotides break off and are lost during transcription process. No, so it is A. Use the diagram below and the information to answer 99 till 101. This is RNA processing. Okay, so DNA to this is the one when you make primary transcript. This is called as transcription. When RNA primary transcript is converted into mRNA, this is called as RNA splicing or RNA editing or RNA editing. Okay, RNA splicing, RNA modification, so it's also called as modification. So the better for you is RNA splicing, let's say. Okay, so we need to see what happens. So complete the table below to say what is happening in each labeled process and its role. Number one, so what is happening in the number one? We said it is transcription. So what's the role? Copying instructions for the processing of RNA. Number two is in number two, what you see, GTP cap is added and poly A tail is added. And in number three, this is the spliced exon, while these are removed, so you say introns removed. This is what happened in number three. And why this happened? So that you can have only final mRNA. Better please refer to the IPQs. Okay, alternative splicing allows single gene to produce multiple proteins. Describe how this process occurs and explain significant in increasing. Of course it occurs because when the introns are removed, the exons can join together they randomly, and they may lead to formation of different sequences and lead to formation of different proteins. And this process, what is the significance? It increases the protein diversity. So of course it leads to protein variety, so it leads to diversity without requiring more genes, without requiring more genes. Okay, a mutation occurs in a gene leading to defective splicing. Predict how this could affect the final mRNA transcript and the resulting protein. Of course, if there is a defect in the protein, it will lead to like different introns, different exons, and it will lead to incorrect splicing sites and lead to different protein, altered protein. So its structure will be different and its function will be different. Okay, complete the table below to compare the transcription in eukaryotes. We did it already. Where does it occur? Nucleus. Does RNA processing takes place? Yes. Where does it occur? In cytoplasm. And does RNA processing takes place? No. Complete the table below to identify the post-transcriptional modifications, and this is the same terminology we use for RNA processing as per each listed. Removal of the non-coding regions, introns, and joining of the coding regions. So remember, the coding regions are exons and non-coding regions are introns. Post-transcriptional modification. The process name is RNA splicing, RNA processing, RNA editing, whatever you want to say. Sequence of the repeated adenine bases that facilitate export to mature RNA from the nucleus. So this is adenine like A, so it is poly A tail added. Okay, and poly A tail is added at 3 prime end, at 3 prime end, remember that. Also G cap is added at 5 prime end. Differentiate between the introns and exons, next one, and this is very clear over here. Introns are non-coding regions and the exons are the coding regions. The white gene of the fruit fly is 5A. So this is a repeated question possibly. We did it up. It was this one. It's the same one. Yeah, we did it up. And also this one, we did it up. Yeah, we did them here. Yes, these are the two. The gene contains and many non-coding stretches. So we will do many non-coding stretches. Yes, this one. Which of the following correctly differentiate between exons and introns? We already said exons are the coding, non-coding, wrong. Exons and introns both code, wrong. Exons are the coding sequences in the final mRNA, while introns are the non-coding sequences that are removed during RNA processing. Yes, exons are only found in the prokaryotic. No, so C is correct. Moving to the next one, identify the function of the non-coding RNA molecule. So there are two non-coding RNA molecules. Which one is the coding RNA? Think. What's the coding RNA? mRNA. So rRNA and tRNA are the non-coding ones. Which type of RNA is shown in this diagram? This is a loop, pin shape, and also you have this one that is an anticodon. And over here on the top over here you have amino acid. Okay, you have over here amino acid. So this is a tRNA. Which number in the diagram represents the part that contains amino acid? Of course, number 1. We already did it. Now complete the table to identify the function of each listed. We just defined them up. So how did you define it? You can say it now. mRNA. So what does mRNA do? It carries the genetic information. It carries the genetic information from DNA to ribosomes. Okay. rRNA, it forms better we properly say it, for example, ribosomes. rRNA, they are component of ribosomes. Okay, and tRNA, they carry amino acid from the diagram above, amino acid and anticodon, and these anticodons are complementary to the codons on the mRNA. So this mRNA contain codons. Okay. Oh, great. We reached to the last one. Describe using diagram an overview of the process of the translation. So what is translation? RNA to protein. So this is what we need now, mRNA to protein. Which of the following correctly describes an overview of the translation process? Translation occurs in the nucleus? No. Where does it occur? In the cytoplasm. Translation occurs at the ribosomes? Yes. Where mRNA is decoded into a polypeptide, looks like quite good, but we need to see like what happens. Yes. With the help of tRNA? Yes. Ribosomal subunits and enzymes? Yes. So this is correct. So this is basically the definition of the translation in detail. During translation, ribosomes break down mRNA? No. Translation takes place in the Golgi apparatus? No. The diagram below shows which is related to the process of protein synthesis in the cell. To answer question 111. So this is amino acid 1, amino acid 2, amino acid 3. And what is B? B is tRNA. Okay. And what are these? These are anticodons. Okay. And these three are codons. And what is A? A is a ribosome. And what is C? This is the mRNA. So you can clearly see what are the structures. So structure A, wow, Structure A is ribosome, structure B is tRNA, and structure C is mRNA. Okay, and what are the functions? These two we wrote up already, we have written them up, but the upper one, this is the organelle where protein synthesis takes place, where protein synthesis takes place. Okay, the other two we wrote up. Okay, describe the structure and function of the tRNA. We already saw this is the tRNA. It looks like a loop, and 2 is the anticodon, 2 is anticodon, and 1 is amino acid. We know it already. Okay, which of the following correctly describes the function of the tRNA molecules? tRNA molecule transport mRNA molecules? No. tRNA molecule transport? No. tRNA molecule transfer amino acids to the growing polypeptide chain? Yes. tRNA molecule transmit the protein sequence to the ribosomes? No. So it's quite obvious that the answer is C, that it carries amino acids and the anticodons. We already did this. Label 1 is amino acid, either this or this. No. And label 2 is anticodon, so it's A. Based on the genetic code chart on page 28, so you will be given codon table. Possibly there is a codon table. Okay, there is no codon table. Let's use these. So in the codon table, you will have the nucleotide sequence. So for example, like this G, 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 U, G, C, like this one. Okay, so when you are doing this, you need to read GGC. So go here, GGC code for proline. Okay, so you will say proline, and UGC, there is no other proline, so of course this is answer. UGC, this is UGC, it is threonine. Okay, now remember the rule. The rule over here is, if you are given full names, write full name. Okay, like this one, write full name. But sometime they give you like this: pro, ella, trio, gly, cis, ella, like this. They give you 3 letters. So if the 3 letters are given, write 3 letters. Don't be so like rude, or what should I say, I don't know. Anyways, to write only the first letter. Okay, so sometime during SBQs we have observed like you write only the first letter. So instead of writing P, so you write P dash T. So if you do P dash T, khalas, this is wrong. This is wrong, not accepted. Okay, so as per the table given, if the table says pro dash 3, that's okay. If the table say full name, say full name. So this is CCC. Let us see, we have CCC here. We don't have CCC here. It's okay. Normally it will be maybe this one, glycine. Anyhow, so it's CCC. Maybe it code for proline again. Okay, let's go to this diagram. So this is the same diagram as given above. Identify the nucleotide sequence from 1 to 8. So what is 1 opposite to this? If this is U, it means it was T, right, in the DNA. So U, so it is when you go to the opposite one, you say it is A. I'm not, okay, let's write here A. Then if it is A, then next is U. If it is U, then A. What is next? Number 4, A, U, then U, then C. Next it's C, so it is G, C, and C. Okay, use the genetic code chart again to identify the amino acids that will be coded by these. Okay, now very important over here, you will use these. Okay, because these are the codons, and they are saying use the which chart? Codon chart. Okay, you are not gonna use the anticodon chart. So some students made a mistake during SBQ, that when this question was given, they used these to write answer. No, that's totally wrong. So you need to use the codon table, this one. So you will find what does UAU code for. So you do this one, and above that, remember 3 more informations. AUG is start codon. Okay, and that always code for what? Methionine, meth. Then UAA, UAG, and UGA. These three are the stop codons, which doesn't code anything and it leads to the termination of the translation. Possibly the last page. Describe how genetic information in retroviruses is a special case because it has reverse transcription, and that we studied already. Transcription that happens with the help of an enzyme reverse transcriptase. So who do this? It is happening with the help of reverse transcriptase. Okay, so this diagram we labeled up, you should be knowing it. Describe what is happening in the process labeled X. It is reverse transcription, and predict one situation in this process occurs when the organisms they have their genetic material RNA, it occurs to convert into DNA, and then so on. Which of the following shows the flow of information in retroviruses? So we know that retroviruses have RNA to DNA, then DNA to again RNA, and then RNA to protein. So you can find this one, that's B. Which of the following enzyme? Reverse transcriptase, right? And here is a big good luck. Okay, thank you.